Hey guys, welcome back. We are moving on to discussing cell-free systems. And of course, the name would imply there is no cell. So we're a cell biology class, but we're now moving inside the cell. Okay. We've talked about fractionation of parts of the cell. So once we fractionate, once we separate uh, the materials, then we can then try to make that process happen in a test tube. Because once we do that, we're free from any kind of noise, any kind of lot of variation. We can control exactly what's happening in that test tube, right? So it's a reconstitution process that we're doing, and we have no cells. And the first example that was probably in the 1980s was in vitro transcription translation systems. So you could take a gene, put it in a test tube with the required materials that makes a messenger RNA, and then you put ribosomes and the materials for a protein, and we could synthesize proteins directly without putting any genes in a cell, not making any kind of mutant kind of cell lines. We could just make the protein straight in a test tube. And once you can do that, then you're talking you can scale up. And so now a lot of the biotech industry and whatnot, we've scaled up from a small amount, and then you can do a big vat, a whole fermentation tank, a whole production tank at that point. And they isolated the materials from to do plant work from the wheat germ. Seeds, you can imagine, need all the materials to, once a seed germinates, it needs all the ribosomes and materials to get the plant growing. And the same things we found, rabbit reticulocytes were also broken apart, and we were taking the transcription and translational materials out of those, and you could store them. -80. Theoretically, you can make a kit and sell it. And so, if you go to the biotech Walmarts there, Sigma Chemicals, Promega, some of these other companies, they will sell you this, and it will be sent on, frozen, and you can thaw it out and start the process at the right temperature. It would have all the kind of like a cake mix. It has all the ingredients there. And so again, we did, somebody had to fractionate from the cells these materials. Okay, so when we talked about all the different centrifugation and all the processes there, that was the start of being able to get these materials, organelles, and things in a test tube. And so in the cell-free system, we're going to talk about uh, that process here. And these fractionations were the ultracentrifugation or the separation technique. We could study then freely from all the side reactions that might be occurring in a cell. So we take the actual process we want to focus on and step away from anything else here. And so that's why a cell-free system will be widely used. And so where we're going in the future will be a synthetic biology. What they will do is not just organelles now, they'll put pathways, and actually from the materials, they'll build a cell. So we've talking about taking a cell, breaking it apart, and using those materials to make proteins. Well, the synthetic biologists will kind of start bottom up and make cells now that only have what we want in it. So we're maybe 10 years away from that, maybe sooner, that we'll be having to make our own cells with materials we want. So if you read about synthetic biology, in some ways that's kind of been the table set, because we did the from the cell, broke it apart and did processes, and now synthetic is we're going to take those processes and put them back in. But again, we decide what's in the cell, so you'll have completely artificial type cells that will probably can never exist in nature. And so with all the DNA cloning and materials, getting the Human Genome Project, uh, it's kind of hard to do in vitro studies necessarily. It's expensive, certainly, and again, there's a lot of noise involved. So the in vitro systems uh, were first developed again to look at proteins, and you can manipulate the conditions to make, it's called translational machinery. In the extract, we can change the temperature, pH, ionic strength, and change the environment. And so we don't have to deal with having to work with cells that can be temperamental. Now we're in a test tube here, and we can produce proteins that in fact could even be toxic. And so now if they kill the cells or make them sick, we can take that directly out and put that in a host organism as well. So the disadvantage until probably the last 10 years where it was pretty low efficiency of making proteins, but a lot of that has gone away now. They've really had plenty of time to increase the efficiency, and so that used to be more of a problem probably in the 1990s than it is now. So again, in vitro means outside the cell, and usually the kits you get are coupled. In other words, they'll have the materials to make the messenger RNA and make the protein. So you can use any gene. We can make any gene. Maybe there's a hard to grow cell type. Okay, uh, we can now use this system to study the proteins in that cell, and we don't have to spend a lot of money to try to manipulate a finicky system to grow here. So we can make proteins and look for other proteins. They call this labeled probes, so we can actually tag the proteins and figure out what they may bind. And also we can make a lot of messenger RNA if we needed that. And you're saying, what is that for? Well, we probably won't talk a lot about this until molecular, but people use that for microinjection. They'll actually take a cell and put messenger RNA into it. So we'll manipulate a cell and put a special messenger RNA for a protein that doesn't exist in that cell. And we could put a viral protein inside one of our cells, for example, and see the effect when you get infected with a virus, right? Because if you're in a lab that's working with a virus, you probably is class 2, you're going to have to do a lot of care, right? You don't want to infect yourself. Versus you just take the viral protein gene and put it in and see where it goes inside its host cell, and it's a lot more safer that way. So the translation side, we can express a lot of proteins rapidly, and those proteins could have medical purposes. Almost all the insulin now given to diabetics type 1 is recombinant. They call it recombinant. That's from the gene human insulin, and it can be made in bacteria. So you can just grow large amounts of, like I said, tankfuls of that insulin protein. You probably don't know this, but probably in the 1960s and back, a lot of the insulin actually was from horses or other livestock. And so there are certain diabetics that their system doesn't like a foreign insulin, and you would have an immune issue. So there was issues with getting insulin into those people. Now we have human insulin in humans, and so it works quite well. So that was a big breakthrough. Growth factors, human growth factors. Some people were born with low levels. You can then uh, uh, give those give those infants and whatnot, and try to stabilize that. And there's other medicinal proteins as well. And again, cell-free means there's no organism or cell as the host for the process. And this is a cartoon just to kind of show you aspects of it. I got two or three of these. So here is a gene that's called a template. So template means that's something kind of like a Xerox. You go to the Xerox machine and you put your whatever you're going to copy. Well, that's your template, right? And the copy comes out. So if you think about this, we're putting the original gene and we're getting the copy of the protein that comes from that gene. You see the protein right here. So think about the big picture here. That is what we're doing. So we're going from these processes to finally get to this protein. And you're saying, well, why don't we just break the cell open and get protein? The amount, imagine this protein is rare. And so now with this system, we can grow a tank full. I mean, we're talking literally gallons of material that has the protein in it. So we can study it, look at its structure, figure out all the, if it's a mutant protein, does it cause a disease? We can study all that, have plenty to work with. So the companies will sell you this frozen mixture here, and inside it will have an energy generation system, because you got to have some ATP happening. It will have nucleotides. It's NTPs. That's AGU. I say AGU and C. Is that right? The U to the T. I forgot. Anyway, you could figure that out. But it's your cell there. We have to have an RNA polymerase, and you finally get a message. And by the way, when you're doing this, if you want to ask the question, does transcription require energy? Just don't put this part in there and see if you get a protein. You won't. Do we need RNA polymerase? Well, don't put it in there and see what happens. Again, you won't get a protein. So this is kind of the output that you can measure, okay? And so then also in this mix are ribosomes, and ribosomes have other proteins, translational factors. They're amino acids, tRNAs, and energy system that requires the ribosomes to go from the AT site, and you can make the protein there. And so that's called, of course, you know this translation, and we now have a protein. So we can make any protein we want from any gene. Once we have a gene, we can make a protein. And again, if it's a toxic protein or from a very it's a low express protein, we can make plenty of it and study it using physical biochemical techniques that we'll talk. For instance, protein crystallization is one. We can actually freeze the structure and figure out exactly what's the nature of the protein is. Okay. So here's another one, just show you here, there are prokaryotic systems, there are eukaryotic systems. And here's the cartoon. We get the cell lysate, so we break the cells open with sound waves, freeze thaw. I think some of the methods we talked about in our fractionation scheme. And look, we get the translational factors, the ribosomes, the tRNAs. Here's our template. We get a messenger RNA from that, and then we add the amino acids, energy, other kinds of supplements, and we put that all into a test tube, and that's called de novo, or brand new synthesis. And in this case, looks like they were making antibodies or antigen fragments. So that's kind of neat. So when we talked about making, monoclonal or polyclonal antibodies, we have a way now we can make artificial, i.e., or in vitro antibodies for further study. So we can set this as whatever this template is, we can make it here, assuming the gene doesn't mess up and interfere with the machinery, right? If you put something in that would interfere with the machinery, then of course we would not get functional proteins. So this is a prokaryotic system. This is just another cartoon I found. Here's using the extract, the template, and sometimes they call this mix that they'll sell frozen, comes to you in a tube, a master mix. That means it has everything in it, right? So you don't have to add, right? It's like pancake mix, just add water or add milk. Well, that's the same thing. This is kind of the protein pancake mix. We just add the cell extract and the DNA, and there we go. And bacteria do best in a test tube at 30 degrees, and that gives us a stable expression in a tube, and we end up with as much protein as we have master mix from. And it just shows that. And we've talked about the energy source, translational, other proteins. So there's other proteins involved in translation. You may not have hit that too hard. Obviously, we need the small and large ribosomal subunits and the tRNAs, and that's all there in the mix, and you get protein. And here's somebody was paid. Look, they copyrighted TNT. Boy, that sounds dynamite, doesn't it? So it's a master mix. It contains all the components for a coupled transcription translation. We add the gene, or when in molecular, we'll talk about amplifying genes via PCR. And if you ever wonder we could take a fragment of a gene and actually express part of a protein, like a domain. Okay, yeah, we've been talking about domains. So people say, well, how do we know a domain is important? Well, you can actually make just that part of a protein in these in vitro translation transcription mixes, and we can have then a partial protein here for further study. Does this domain interact with other proteins? Does this domain bind to DNA? Is it an enzyme? Does it bind to a substrate? So look at the questions we can address here. And it takes about an hour and a half. So that's not bad at all for time-wise to get this done. And here's just another cartoon breaking the cells open, but I thought this was neat. So now you can do multiple genes, and they call it a gene network. You put that material with the extracts, with all the machinery, and you put it on a paper disc. You freeze dry it -80, take all the water, and then theoretically you can take these discs and rehydrate, and then there may be some other kind of chemical or other trigger, and you can get proteins made. And so now we have a synthetic biology based little disc that can make a protein if you needed to. So that would be kind of neat, that if you didn't want to isolate the gene, and just you could order these tabs and make your protein right there, and then you could purify the protein from these discs by fractionation, kind of in the next thing. So we will talk about protein fractionation coming up. Okay, so just kind of put this in here to remember we could take the protein. I guess the purple is the ones that made the protein here.